talking about X chromosome inactivation. I deeply and sincerely apologize for not having put the video I assigned a couple of class grades ago on YouTube. Maybe some of you got a chance to watch it this morning. That's not critical, and it didn't have anything to do with the Socrative Quiz today anyway. But I was hoping that people would have that background for today's class. So again, apologies. We'll talk about X chromosome inactivation, sexual orientation, and asthma we'll talk about next class. These are 3 case studies in epigenetics, how environment might affect your traits, your phenotypes. For those of you that saw the video, understand why I put this picture of a calico cat on the introduction slide for X chromosome inactivation. If you haven't watched the video yet, that's fine because we're going to run through a brief little interlude right now that's going to review all of this material. So watch a quick video. Yeah, I know. We have movies in class. Yeah. Better than movies out of class?
Is the final included in that drop the lowest?
Good question. The final exam, so you drop the lowest exam grade. The exams are the midterms. So the final doesn't, if you get a lowest score as the final, that doesn't count. It's the lowest of the first 3 exams that will get dropped. Thank you, that's a good question to ask. Okay, so here's a bit of introduction about X chromosome inactivation.
Female with 2 X chromosomes. The cell divides, and the embryo is getting slightly bigger, and still, each cell has both X chromosomes active. But in early embryogenesis, each cell will inactivate one of its X's, and one cell will remain with the paternal X as active, while the other one, the maternal one. And now, this process will happen at random, and you'll have almost half of the cell with the maternal X active and half of the paternal X. The embryo will continue to grow, cells divide, all the descendants of that cell, all the descendants will keep the same X active. And you've seen this in calico cats, right? The coat color gene is on the X chromosome, and that's precisely why these cats look like this in mosaic appearance, because of the process of X chromosome inactivation.
Short and sweet. So that's X chromosome inactivation. Which sex does this process happen in? Females. Does it happen in males? No male calicos? So the reason that males don't do X chromosome inactivation is we only have 1 X chromosome to start with. And the whole reason, not that there's a reason because evolution doesn't have a brain, but the reason, the benefit of having X chromosome inactivation happen in females is because, as I described in the video for those of you that watched it, we need to have an equal amount of X chromosomes between males and females. And there are 2 ways to do this, and different organisms use different approaches to do this. So in humans, we have X chromosome inactivation in females. So 1 X chromosome becomes, what is that blob that I just drew? It's a chromosome. We call it a bar body. That's a B-A-R-R, if you can't. Otherwise it looks like maybe Klingon. What chromatin structure is it in? Is it euchromatin or heterochromatin? It's heterochromatin. The entire X chromosome is condensed. It's really, 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 really highly condensed. And that's based on epigenetic change. So in this case, the way that females turn off 1 copy of their X chromosome in every cell is through methylation. So chemical modification turns off 1 chromosome at random. Every cell different, 1 X chromosome or the other. So that makes, in humans, the female genotype essentially functionally like the male genotype in that female cells have one X chromosome that's producing transcripts and males have one X chromosome in their cells that are producing transcript. Yeah?
Would it accidentally for the male, the Y gets compressed?
So males don't do this process at all. Yeah. So only females do X chromosome inactivation. And it's because we're the, oh, I hate to say this, guys, we're the inferior type, I guess. We're lacking an X chromosome, so the females are nice enough to us to make up for it by turning off one of their X chromosomes. We don't do anything, females compensate for that. In humans, and this is different in different species, yeah.
What about male like Klinefelters?
Ah, so fantastic question. What happens if you're a male and you're XXY? Klinefelters says that you're male, but you've got two X chromosomes. I don't know if X chromosome inactivation occurs. I encourage all of you to try to find that information on your own. You can Google it. That's what I'm going to do when I get back to my office, and I'll try to report back to you next class with an answer.
When I couldn't find your video last week, I found another video on calico cats and X chromosome
Okay.
activation. He's the guy, in his knowledge, he said that likely a cat with Klinefelter syndrome, you could technically have a male cat that would be calico.
Okay. So it might, yeah, I'll have to look into it.
But super rare.
Yeah, Klinefelter cats. The one exception to this, or the exception I know about, is Drosophila, fruit flies, which we know a lot about. Remember, they have XX and XY organisms too, so I'm going to color these blue instead of black for humans. We'll do blue for Drosophila. In Drosophila, the females don't do X chromosome inactivation. So what do males have to do to compensate? In male Drosophila, their X chromosome produces twice the amount of transcription as female X chromosomes do. So instead of females having the amount of X chromosomes they have, which is what happens in mammals, in Drosophila males, they double the amount of gene expression from their one X chromosome to compensate, so that males are like females, at least in the amount of transcription that they produce from their X chromosome. So there are many different solutions to this problem that different organisms have come up with evolutionarily, but we're focused on the one that has to do with epigenetics, which is the X chromosome silencing in females. And the critical point here on this slide is simply this high histone methylation and DNA methylation also associated with the X chromosome. So methylation, hopefully you will remember this. Methylation is usually associated with silencing or inactivation, heterochromatin. And what's the modification that's usually associated with euchromatin? Acetylation, right. Now, this is not a hard and fast, 100% of the time true rule, but it's pretty close. So in general, methylation, silencing, acetylation, active transcription. So we saw a picture of the calico cats. Here's a fancy artistic bust of a lady, both of which kind of demonstrate the same idea, that random parts of the body have one X chromosome or the other X chromosome turned on. Calico cats, that has to do with pigmentation. Maybe there are human females walking around that have different pigmentation versions of different alleles on their two X chromosomes, and you would actually have this sort of patchwork phenotype. Maybe, I don't know. What dictates how big the patches of orange or black fur are on calico cat females?
The stage at which the silencing occurs?
Right. So that was the initial part of that movie I just showed. One cell becomes two cells, and again, it's on my movie as well. One cell becomes two, two becomes four. So the stage of development where the X chromosome inactivation happens dictates how big and how many of these spots there are. So if X chromosome inactivation in a calico cat happens at the 2-cell stage during development, where one cell turns on, only has active, say, the black pigmentation gene, then you'd get a cat that's 50% black. But if this process happens at the 4-cell stage, that's not 800, by the way, that's 4 cells. And one X chromosome randomly is inactivated and produces only black pigment, then you get a 25% black animal. So you can kind of guess, you can look at an organism, you can sort of infer how far into development X chromosome inactivation occurs. Is it the 2-cell stage, the 4-cell stage, the 256-cell stage, by how big the patches of coat color are, for example, on a tortoiseshell cat? And I want to take a brief pause here and take a look at some of these responses from your Socrative activities. So put epigenetics on hold for just a second, because there's at least one important point to be made here. So I want you to know how you stack up related to the rest of your classmates in terms of your opinions about whether or not, for example, all foods that have been genetically modified should be labeled. So 60-40, pretty even. Correct answer, there was no correct answer. In Socrative, I can't, if I do a true and false, it forces me to say that one of them is correct. So there is no correct answer. Sorry. So this is the important one. That one's, I'm happy to know your opinions, but I want to know specifically, this is a vast change from the first time you took this Socrative quiz. So right before we started talking about GMOs, I got your input before we talked about them in class, where these numbers were basically flipped. Most of you, well, it depends on who's in the classroom right now and who is taking this quiz. Most students that responded the first time wanted all foods with DNA to be labeled.
Doesn't all food contain DNA?
So that is the important question. Doesn't all food contain DNA?
So you have to label your apples?
So everything would be labeled. Prove me wrong. So there are foods that don't contain DNA. Can anybody think of any? We've got a couple minutes. But it still contains DNA, so like wheat products came from plants, so bread contains DNA, cereals contain DNA, beer's got yeast, and grains, it contains DNA.
Mineral seasonings like salt.
Salt. One of the few things that you buy at the grocery store that does not contain DNA. It's just a chemical. Salt. Anything else? Grocery store? Water. Baking soda.
Those aren't like.
But sugar came from some sort of organic. So whether or not we've had in past semesters, we've had debates in class about whether or not sugar does probably have some residual DNA from the plants it was isolated from or not. I mean, sugar could be GMO, so you might have to label it separately. I just wanted to point this out because it's important to know that all of our food contains DNA, pretty much. And this replicates, in case you're curious about this and want to bring this up at the next cocktail party you're at, every year, there's a university in the Midwest that does a public attitude survey about food materials. It's a land-grant ag school as well, and they do something like this. And every year they ask the same question, basically, to the public, not to students, but just they send it out, normal survey of the public, should DNA containing foods be labeled, and the public basically says the same thing. Yeah, everything that contains DNA should be labeled. Yeah, I contain DNA. Maybe I'll print t-shirts for everybody. So please, if you're interested in helping out the cause of increasing public understanding of science, maybe strike up this conversation with your friends, family, whoever. Let's check out that last one too. So, could consuming a GMO pose an immediate health risk to humans? Thirds say they think so, 2/3 not. That's a bit different from the first time I polled you on this question. So attitudes in class seem to be shifting. I'm not going to say which I think is true, but things are changing. Yeah.
My only question on this one is like how we talk about growth hormone and stuff that we don't necessarily know right away.
Fair enough. Yeah.
What it's doing right now. Like it could just be the growth hormone's completely. It doesn't do anything to us. It actually has no effect.
Right. Yeah, it does make it sort of hard to answer this sort of a question because it's very vague. Which GMO are we talking about? Which approach are we talking about?
The presentation last Friday of Dr. Professor Paul Maselli,
Right.
I answered false because I felt like if he would feed it to his kids, then he does the research on this himself.
So yeah, if an expert says it's okay, then maybe it's okay.
Maybe.
Maybe. That's not necessarily always the way to go, because one person's informed opinion can still be wrong.
Most of these foods actually have to go through the USDA first, so before it actually makes it onto shelf.
Not necessarily. Yeah, unfortunately.
Did you say that the type of potato that was like avoiding people, that was conventionally bred, right?
Right.
And they
So yeah, we've got the conventionally bred potato that fortunately wasn't fed to anybody that had high levels of solanine, which is a toxin.
When did that happen?
That was in the 60s or 70s.
Okay.
It said 80s.
Was it the 80s?
Because I feel like at this
Yeah.
point, nobody would put out any food that would be test just because of public,
So traditional breeding is not always safe, but that would be labeled, depending on what your definition of GMO is, your personal definition. If we had all foods be labeled GMOs, if you include traditional breeding as a genetic modification, then that potato would have been labeled. Not that that would have helped anything, but pretty much everything you buy as produce at the grocery store at this point would be a GMO.
Even the salt you're talking about has been tinkered with.
Yeah.
So if we're talking about like antibiotic feeding in like poultry or something,
Mhm.
I mean, is that genetically modifying anything other than the bacteria, or I mean, how is it that the chicken gets so like plump, so to speak?
Either we select traits for that, or we pump them full of hormones that allows for. better
Right.
storage.
So use of antibiotics gets beyond genetic modification, fortunately,
Epigenetic?
but maybe epigenetic. I'm not sure if there's any data to support whether or not antibiotic addition to feed affects epigenetics, but maybe a ripe area for new research. Here's something we do know about epigenetics. If there was a gene that controlled sexual orientation, just one, we would have found it by now. Just because of the amount of societal interest in figuring out is there a genetic basis or not to sexual preference. So, there is no quote-unquote gay gene. But there are still people that are interested in knowing whether there's a genetic component, predisposition to sexual preference, what have you. So the question that I'm going to show you a little bit of research about was, could there be a genetic aspect of sexual preference, sexual orientation, specifically with regard to epigenetics? Chemical modifications to histones or to DNA itself that change the way genes are turned on and off might control something about sexual preference. Now, take this, speaking of salt and foods that contain no DNA, take this research with a grain of salt. So even though I'm going to show it to you afterwards, we're going to have a little bit of a public dissection about whether or not we believe this research is valid or not. But this was presented in 2015 at a professional conference to my knowledge, although I haven't looked in the last year, I'm not sure that this has actually been formally published in press, that is, in print. But this research got a ton of media coverage, not surprisingly. Said that they had a simple genetic test that could predict whether or not you were homosexual or heterosexual, right? These are some of the headlines. And this is something that geneticists and all biologists really, and all scientists for that matter, have to struggle with all the time, is the fact that media uses sound bites and simple headlines to get our attention, to make us read the media or watch the video. It's especially bad with, probably shouldn't use my opinions, but it's especially bad with the web because all of these sites get paid by, a lot of these sites get paid by number of clicks, right? You click onto the page, then all these advertisements show up, and that's how the people that run the web pages get money. So it's kind of inherent now in media that you want to generate click-throughs, so you write clickbait-like ads. One weird new trick to figure out whether or not you're straight or gay. So that you come up with these sorts of headlines, like saliva test can predict male sexual orientation. It's not as simple as that, but that's how the headlines are written. They're written simply. Researchers say gene changes show who's gay. So here's what these scientists did, in brief. There's more detail, of course, than this, but these are the highlights. They took male twins. Twin studies are great for genetics because you have two individuals that are genetically almost identical, better than you can get by just looking at siblings, so pairs of twins. They have 37 pairs where there's one homosexual and one heterosexual, and 10 pairs where both of the twins were homosexual. And they want to look at epigenetic differences between the twins. So you extract DNA from both of the twins, and you look at methylation and acetylation and all these other chemical changes that occur to histones in the DNA, and you just look for a correlation. Does the homosexual twin have a certain pattern of changes to their DNA, methylation marks, acetylation marks, compared to the heterosexual brother? And there's the result. There were 9 chromosomal regions where DNA methylation, which we heard about at the start of class, DNA methylation, 2 different nucleotides get methylated, but what's the most common one? This is from a while back in class. There's a particular dinucleotide, 2 nucleotides together, where the cytosine gets methylated. So you get methylcytosine when the cytosine is next to, there's a phosphate, and then there's a guanine. So it's CPG, which, again, that P in the middle is just the phosphate that links the C and the G. So CG dinucleotides tend to be a spot where methylation of the cytosine happens. One of these 9 regions includes an immune gene, major histocompatibility complex class 2 gene. Another one of these 9 regions contained a gene that has, we don't know what it does specifically, but it has something to do with neuronal function. The other 7 areas of the genome, we just don't know what the genes in those regions do. That's what they found. So statistically significant association between epigenetic changes and sexual orientation. 9 different regions of the genome. 9. So has anybody spotted any potential flaws, holes, issues? Do you have any questions about how this was done? Go ahead.
Kind of interesting that I know it's about homosexuality, but they took twins where both are homosexual, one was homosexual. They didn't do any where neither of them were homosexual, and I can see why they didn't do that, but at the same time, if they're looking at twins where one's homosexual and one's not, and they say, well, this twin has just this little bit of difference from the other, so that might be why they're different. But if they took 2 twins that were both heterosexual and say one was a little different than the other, then they could use that against their information.
Fantastic. So we're missing basically the 3rd possible experimental group, a control group. Where were the pairs of twins where both of the twins are heterosexual? Great question. Yeah.
Sample size?
Sample size. What was the number of twin pairs?
37 where one
Yeah, 37 plus 10. So that's, and then there are some other questions beyond that, like, go ahead.
You say gender? They just focus on one gender?
Why only males? Why not females? I mean, 47 pairs of twins, they happen to all be male? Probably not. They probably chose to study male twin pairs, not both sexes. Why? That should start ringing alarm bells. So is this going to be a pattern that's true for all humans, or is this something male specific, or is it worse? Is it just artifact? It's nonsense.
Maybe even the age range to see what level of epigenetics over time changes.
Sure. So there are many things about age. They didn't tell us, well, at least I didn't just tell you about the ages. You'd have to look more into the research to find out if they report the age range of these twin pairs. But there are a few issues there. One is, yeah, epigenetic marks can change over time. So you'd want to know where all of these twin pairs in their 20s, was it 20s through 70s, teens through, I don't know. But that could definitely impact the results, the interpretation. Yeah.
We don't know the environment that they come from.
So environment probably has a huge impact. And of course, in genetic studies, it's really hard to control for environment. That's why we use twins. But we want to make sure that the twins were twins that were raised in the same household, probably, for example, went to the same schools, were in as similar an environment as possible. That's usually why twins are chosen for these sorts of studies. Yeah.
I don't know how to protect it, but on the other page where you're showing the headlines, there was like an Indian flag and another country's flag for maybe nationality.
Yep, we don't know race, which may or may not have something important to do with this process, but it could. So you want to know, were all of these pairs of twins from the same city? Because then they would be potentially totally not representative of the populace in general. So they might be making conclusions about a specific ethnicity if it was in a particular city that has mostly one ethnic group. How were they proposing, if you remember those headlines, how were they proposing to collect DNA to test for your sexual preference? Saliva. Saliva have anything to do with your sexual preference? No. Oh, I don't know, maybe I shouldn't ask. One issue that was raised by others was, well, what does saliva have to do with sexual preference? The epigenetic marks in the DNA in your saliva might not have anything to do with epigenetic marks elsewhere in your body that might be more important in controlling sexual preference, like maybe in your brain. The different parts of your body can have different epigenetic marks. So who cares whether or not your DNA in your saliva is methylated, for example, if that's not the part of your body that's controlling sexual preference? Maybe you want to take brain samples from people and test. I mean, that's why they didn't, of course. It's hard to get people to agree to give brain tissue for these sorts of studies, but it's an important question. Are they looking at the right tissue type to be able to answer this question? And there are probably many more concerns as well. Now, for the interest of time, I'm going to skip this slide. We're definitely coming back to it next class. And next class, we're also going to finally get into asthma and the effect of breathing polluted air on histone modifications and our kids. Last thing for next class, so no movies to watch, no readings. We're now in the free fall part of the semester where it's just all ad hoc, ad lib. We're talking about things that are interesting. By the way, if you've got topics that we haven't talked about yet that you'd like to hear about, email me or tell me in person. I'll see if I can work them in. The one request I do have for you for next class is to go to Blackboard, go to the URLs folder, click on that top link, our box folder, go view what people have been dropping in there for their videos for Comedy Genius, please. There's 3 of them. There might be more now. When you hit that link, log in with your Fresno State user ID and password so that you can leave comments. Please leave comments. Please view these videos of your classmates. If you like it, leave a comment. Again, the video that gets the most likes in terms of comments will win a prize the last day of instruction. See you next class.